All right, well, thank you very much. It's really a pleasure to be here and actually to talk to this department again, because I haven't actually seen most of you, it seems. And I haven't had a chance to talk here since my job talk back in December of 2010. So it's been some time, and I'm happy to report that a lot of studies that were just percolating in my mind have actually gone into press, and I'm happy to share them with you today. Some of you who were at my job talk might recognize a couple slides, but maybe not, and that's all the better. So today, what I'm talking about are large-scale brain network interactivity and aging. Really, I'm not sure how far I'm going to get through this talk, so I'm just going to thank right up front my collaborators, in particular Dan Schachter, who was my mentor and postdoctoral advisor at Harvard. He really kind of I think I pushed him well past his comfort zone with a lot of these studies, and I'm really grateful to him for really believing in me and really allowing me to have the flexibility to see them through. In addition to my other collaborators, Jorge Sepulcra, who does amazing stuff analyzing brain networks, in addition to Dale Stevens and Gary Turner, who I've known for over 10 years now. I met them in my first year in graduate school, and we're still very good friends and very active collaborators. So it's great to still be working with people, and the people that you know in the room. You may very well continue to be working with them throughout your career. So just to give you an outline of my talk, you can see I feel like it's a little ambitious, and it's fine if we don't get through it all. I realize that people who do cognitive neuroscience are more the exception than the rule in this room. Just out of curiosity, can I see some hands for people who are familiar and comfortable with brains? Okay, great. So, and what about cognitive neuroscience in particular, looking at fMRI? All right, so that's actually better than I thought. That's great. So what I'm going to do initially in this talk is to just introduce some really basic concepts and methods with regards to human neuroimaging using fMRI. And this looks at, I'm taking this phrase, two views of brain function, from a paper that Marcus Rakel wrote in Trends in Cognitive Sciences a couple years ago. And this really pulls apart two aspects of the signal in brain imaging data from fMRI. Now, the one that really dominates the field is looking at task-evoked brain activity. And this is what we know as kind of the bread and butter of cognitive neuroscience, what areas of the brain light up when you do something. And there's an entirely different way of looking at the brain in that regards, looking at the spontaneous activity, spontaneous oscillations. And I'll be talking about the distinction between these two approaches. And I draw upon both of these methods in assessing brain function. So next, I'm going to move into the actual studies that I performed, looking at planning as a behavior that drives large-scale brain network interactivity in both young and older adults. And also looking at more specifically about how particular regions of the brain can interact with each other, how these networks are able to talk to one another. And when I embarked on this study, I really just ran smack into these two big fundamental questions that I'd never really paid much mind to. And that was, what is a brain region? And what constitutes a reliable connection between them? And as I'll show you later on in the talk, these are really not trivial matters. And there's a lot of issues and discussion and debate within the neuroscience literature regarding these. And if we get to it, we can talk about some work that I hope to do here as well as part of the future directions. So just to get started, in most studies of human neuroimaging, people will lay in the scanner and engage in a number of different tasks. One standard type of methodology is to have a block design. This is where subjects will be presented a number of different trials over a certain interval of time, followed by a block of fixation where they're just passively resting in the scanner, and then another block of activity where they're performing some task. In this case, it could be an odd or even number detection task. And all of these trials are they're built into a regression model using the GLM, where the activity across these trials are averaged together, and you're looking at the difference in brain activity between performing the task and just simply resting. And on one side of the equation, you just see brain activity associated with resting. On the other, you could see some sort of activity associated with numerical processing. In a similar type of task, this is called a rapid event-related design. This is where these trial types are interleaved much more rapidly with one another. You'll be presented with a fixation followed by some rotating checkerboard, which will drive primary cortex kind of wild, followed by fixation blocks, and then checkerboards again. And once again, it's a similar process whereby a regression model is built and trials are averaged together associated with task or fixation. And you look at the relative difference between them. Now, this relative difference is usually a change in bold signal, which is the signal we get off the MR scanner, a change in percent signal from anything from 0.2% to 3%. So we're talking about like very small effects inside a very noisy type environment. So we're looking at very small effect sizes, very small differences in brain activity in order to localize brain function. Now, the reason we do all of this averaging is because the brain is actually constantly engaging and disengaging all these various patterns of activity that seem quite random. It's engaging in all this spontaneous activity that's happening underneath performance on any type of task. And in fact, for the longest time, this spontaneous activity was really looked upon as noise, which is why we did all this averaging to try to reduce that amount of noise and improve our signal. But in fact, this noise contains a significant amount of structure. And if we can actually detect and dissociate functional atomic networks by examining how brain regions are correlated in time, by examining these low frequency oscillations across long runs in the scanner. Now, the first study to do this was published by Bharat Biswal back in 1995 and was subsequently ignored for a number of years. But let me just show you what he did. So he had a standard motor task. So he had people doing a finger tapping task, and he looked at brain activity during finger tapping versus not moving, and he saw engagement of the motor network. So this is primary motor cortex bilaterally, as well as a supplementary motor area. And then what he did is he defined a region of interest around primary motor cortex in the left hemisphere. And while subjects were just resting quietly in the scanner, he just observed the random fluctuations in bold signal across a 6-minute scanning interval. And then what he did was he correlated the signal in primary motor cortex across the entire brain. And what he found was that activity in motor cortex correlated with the entire extended motor network, which is shown here just at rest. So these subjects are not moving at all. But what you can see is the same network of regions that are engaged when someone is moving. So it's really fascinating because it's the idea that there's this intrinsic architecture in the brain that supports what is later engaged during behavior. And it took another 6 years before someone even attempted to replicate this method. And this was done by Michael Grekus at Stanford. What they did was a standard visual processing task, and they looked at task-induced deactivations and identified this, which was referred to as the default network. Same areas I showed you before, these are reliably engaged during fixation relative to another active, externally focused task. And if we took, what they did was he took the activity in the posterior cingulate, which is the midline structure here, and correlated activity there with the rest of the brain, and saw, again, the same network that's engaged during, that is suppressed during an externally focused task is associated with the resting state pattern of activity when you're tracking the posterior cingulate. So does this make sense so far? Yes?
What's the time scale of the correlation?
Sure. So a typical sampling rate in the MR scanner to acquire an entire volume of the brain is approximately 2.5 seconds. It can range from 4 seconds down to 2 is your typical range in any given study.
So it's the average over that 2.5 second window is the data point.
Right, that's a single time point. That's right. All right. Now you don't need to know the details of the brain areas involved here, but what has since emerged is that there's actually a number of different intrinsic networks in the brain, referred to as resting state networks, that can be extracted. In this case, this was done using independent components analysis. You can see that there are regions, the structure of this spontaneous activity suggests that you can pull out not just the default network, but any number of visual networks, those associated with control and with motor behavior. And what's quite remarkable is that this pattern of intrinsic connectivity networks is almost identical to what you see during task. So let me just walk you through this slide for a moment. In this study, what they did was they looked at the intrinsic connectivity networks during rest in 70 subjects and compared that to hundreds of studies in the cognitive neuroscience literature and looked at patterns of co-activation during tasks. And what they found was when you have you have visual cortical activity reliably engaged in a number of tasks. This same covariance basically maps on identically to these intrinsic connectivity networks. So this suggests that the pattern of coactivation, the engagement during task, is really the same as what you're seeing in the intrinsic organization of the brain. And this seems to suggest that task-evoked activity might in fact be constrained by the intrinsic connectivity networks. So before we move on, are there any questions about these two potential views of brain function, one relating to activation, another one associated with these low-frequency correlations that depict these large-scale intrinsic networks?
Are there tasks or categories of tasks where you cannot find that?
There are, I'm sure there are, but they're more the exception. So I think it's safe to say that there's not an equipotential for any two regions of the brain to be active at the same time. That it seems to be that there is this biasing towards these underlying networks. All right. So there is substantial evidence to suggest that the brain is organized into these two competing anti-correlated networks. These are two intrinsic connectivity networks, with the default network depicted here in green and the attention network in gold. And these networks support two sort of diametrically opposed functions. The attention network is reliably engaged during any focus on the external world, and it suppresses activity within the default network. Whereas when subjects are engaged in an introspective behavior or when they're passively resting, activity in the attention network goes down and default network goes up. And it seems to have this push-me-pull-you relationship where activity of one suppresses activity in the other. Now, the default network is of particular interest to me because it seems to be supporting what's referred to as stimulus-independent thought. And an example of the type of thought that is autobiographical recollection. So our ability to sort of transport ourselves in time to leave the present moment and to remember something that happened at an earlier point in time in our life. And that engages these core default structures in medial prefrontal cortex, medial parietal cortex, as well as the hippocampus and this extended network. Likewise, the ability to imagine the future or imagine things that haven't yet happened also engage the default network of the brain. And evidence from the neuropsychological literature suggests that this is critically dependent upon the hippocampus. And we see this because patients who have hippocampal amnesia, who are unable to recollect any event from their past, also have this co-occurring deficit where they're unable to imagine anything that could happen to them in the future. Now, for quite some time, I've been very interested in this relationship between the ability to recollect the past and imagine the future, and how it might be supported by this common network. A number of studies came out in 2007 really providing the initial evidence that these two functions were very much linked, and as part of the work that I did with Dan Schachter was to really explore this in more depth. However, I always was somewhat dissatisfied with how the future was being conceived of in these studies. So it was really thought of as episodic simulation, so our ability to ritually imagine the spatial temporal context by which something could happen at some point in time. And just to give you an example of what that would be is, as a target, imagine acquiring a degree. So for an undergraduate, imagine acquiring a degree. If we were to engage in prospection or to mentally transport ourselves forward in time, you can imagine wearing the robes and the hat, and when your row is called up, everyone rising up and going to have the degree conferred upon them. However, I was really interested in a different aspect of how we use the future, and that relates to how we actually plan for the future. So in this context, acquiring a degree takes on a different form. So goal-directed planning for acquiring a degree really involves a number of different steps that aren't necessarily related to ritually imagining that experience, but more in a way that shapes and forms our behavior, such as fulfilling the requirements to acquire the degree, to engage in diligent study habits, as well as avoiding the excesses that can befall undergraduate lifestyles, such as excessive partying and drinking. So in this way, thinking about the future, it takes on a very different shape. And this is something that I was very interested in addressing when I started my postdoc with Dan. But if we look at how planning has been studied previously, it's really been done with these very simple look-ahead puzzle tasks. Now, this one is the Tower of London. It's introduced to the neuropsychological literature by Tim Challis. And in this, he really presented this radical reductionist view of planning. This is where subjects or patients are presented with a goal configuration on this puzzle, followed by a star configuration. And the objective is to figure out the minimum number of moves it takes to solve this puzzle by moving one disk at a time when there's nothing on top. So in this case, the green disk would move down 1, the red disk over 2, the blue disk would go on the green disk, and the red disk would go back. And that would be a 4-move puzzle, which is moderately difficult. As the number of moves increases, more counterintuitive moves needs to be made. And however, it does seem to possess really some fundamental aspects to planning, and that's a recognition of the goal state, as well as the intermediate stages needed to accomplish that goal. And if we image people in the scanner while they're performing the Tower of London task, what you see is robust engagement of the attention network of the brain, these areas in gold. Now, this is perplexing because, as I just mentioned, if you're to imagine the future, or imagine your own personal future and something that could happen to you at some later stage in life, there's robust engagement of the default network. And the question was really, how is it possible that personal planning can engage the brain across these two competitive anti-correlated brain networks? And there is some preliminary evidence to suggest that there might be some sort of intervening brain network that could support, that could facilitate the transfer of information across these networks. And this lies in some studies that isolated function to potentially the anterior insula, which is involved in maintaining that dynamic balance between the attention and default networks. Also, there's part of the anterior extent of the inferior parietal lobule, which is involved in both the control of memory and the control of attention. So it controls both the recollection of internally represented information and also the direction of attention to external space. And the frontal poles have long been known for their role in executive processes that support goal-directed cognition. And finally, there is some very nice some neurophysiological work that using intrinsic connectivities to suggest that there's an extended frontal parietal control network that incorporates all of these aforementioned regions and is topographically interposed between the default and attention network of the brain. But this had really only been observed as a full network during rest and never during task. So in the context of this study, we were testing the hypothesis that this frontal parietal control network could be both coactive or coupled with either the attention or default network, depending upon the task domain. Now, in order to examine this, what we needed to do is to devise a new measure of planning that drew upon autobiographical material. And I did this by essentially interviewing a number of undergraduates, graduate students, and getting a sense of their real-world areas of concern. In addition to determining what their goals were, the steps that they would need to accomplish their goals, as well as a sense of the obstacles. And this procedure was designed to be visually matched with the Tower of London. So the most reliable goal that we had from all of our undergraduates was freedom from debt. So as well as the reliable steps were finding a good job, saving money, and an obstacle to getting out of debt was having fun. So the objective during this task was really to consider the goal state and integrate these two steps and obstacles into a coherent and conceivable personal plan that could be accomplished to fulfill the goal. And just to give you a sense of what this looked like for subjects who were in the scanner, they were presented with the goal state, which you can see, freedom from debt. And then these two steps and obstacle were presented in the disks. And they had 15 seconds to solve this, basically to devise a plan to reach this goal. And then they rated how much detail there was to their plan, followed by fixation. Now, after the scan took place, we thoroughly interviewed all of our subjects about their autobiographical plans, looking at how much they had thought about it before, when they thought they would accomplish this goal in their life, in addition to how novel it was, how much they thought about it, various features. And I've since related that to brain activity. Again, I don't have time to show that today, but there's some interesting associations there. But really, I'm saying it just to convey to you that they really did comply with this task. They were generating genuine autobiographical plans while in the scanner. They also performed the standard Tower of London task. They're presented with their goal state, the full configuration, and they determined the minimum number of moves it would take to solve this problem, followed by fixation. And then as a third sort of control condition, we had a simple counting task where subjects were presented with a random letter sequence within the disks, and they were asked to count up all the vowels and to indicate how many vowels they counted. What's nice about this control condition is that its counting has been used in both the autobiographical memory literature as well as the Tower of London literature to look at relative patterns of activity. Also, by having a common baseline condition, you can see what activity is shared for both of your other experimental conditions. So this study is actually engaged in a number of analyses. My primary focus on it was in the multivariate analysis. I'm not going to share that with you today because it is a little bit more complicated. So what I'm going to show you are some univariate findings that are produced. So to do this, what we did was we had a separate resting state run, and we identified these intrinsic connectivity networks that topographically were essentially here. You see the attention network of the brain. And we looked at the average signal across this entire network and differences in signal in this network for the task conditions. So what we found is within the attention network, there's significant engagement during visuospatial planning and a significant reduction during autobiographical planning. And this is entirely consistent with the idea that this network is engaged during externally directed attention. Now, the opposite of that, we see within the default network that there's robust engagement during autobiographical planning, and there's suppression of the default network during visuospatial planning. And then critically, what we see within this frontal parietal control network, that both planning tasks are robustly engaging it. Now, what's of interest is not just whether or not these networks are engaged, but really, are they coupled? Are they engaged in some sort of cooperation during these task domains? So what you see here is the correlation of activity between these networks during task. On the y-axis is the magnitude of correlation with the frontal parietal control network. And here we see that during visuospatial planning or during the Tower of London task, the attention network activity is robustly coupled with that of the frontal parietal control network. The default network, in contrast, is decoupled from the frontal parietal control network. There's no coupling between default and frontal parietal control during visuospatial planning. Now, during autobiographical planning, the dorsal attention network's activity is decoupled from that of the frontal parietal control network. However, the default network during autobiographical planning is highly coupled with the frontal parietal control network. So what we see here is a task by network interaction. So when one is engaging in personal autobiographical planning, you're not only engaging the default network and engaging the frontal parietal control network, but the pattern of activity between these networks is coupled over time. And this suggests really that the frontal parietal control network can reliably shift in its coupling across these domains, depending upon task demands. Yeah.
If this doesn't make any sense, just dispose of it. But what's the reason for thinking of this control network as a third network that is, sometimes correlated with attention network, sometimes correlated with the default network, as opposed to just the overlap of those two networks?
Let's see. No, I've had this question before from a reviewer, slightly phrased differently. So if I show the multivariate results, you could actually see that much more clearly. That in fact, it's not just that these areas, if you kind of lowered the threshold, you'd see that it bleeds into both, and that's the average across them. These are actually anatomically and functionally very distinct nodes that are not engaged at all during the counting task. But counting is engaging the attention network. So you can dissociate between them, and it's not a matter of just it being that area that's between. Is that fair? Is that
Yeah.
Okay.
Can't show everything.
All right. Did I see another question over here? Or no? Okay. So really what we drew from this study was that it's the planning content that seems to be domain specific, and that's what's driving one or the other of the networks. So for autobiographical, it's driving the default network. For visuospatial, it's driving this attention network. But it's the process of planning itself that's engaging this this frontal parietal control network that's supporting these executive control components to the task. And it's the default activity, when it's coupled with a control network, can support goal-oriented cognition. Now, this is actually a very novel feature of this task, of this study. By and large, there's a general conception within cognitive neuroscience that default network is not engaged or disengaged or antithetical to goal-directed processes. And this is really kind of a legacy within cognitive neuroscience, where people are constantly striving for greater task control and empirical control. So oftentimes stimuli is directed visually. And Goal-directed processes are looked at as like working memory performance, where subjects are manipulating information online based upon what's directly presented in front of them. So tasks that have engaged the default network often are very much unconstrained. And basically, to do these type of studies, you need to release some experimental control and allow participants to generate their own information and bring to the task. And it's very problematic, and fortunately, fewer reviewers are hostile to it now than they were 5 years ago. So it's a challenging area of study, but I think it's an important one. And this study, I think, was really important in showing that not only can the default network be engaged during a goal-directed, personally relevant task, but it's coupling with the frontal parallel control network, which people have long recognized as supporting goal-directed cognition. So it is really this interposed control network that can possibly integrate information from the internal domain and make it relevant to the external world. So in a similar way, this network might actually support information that is generated in memory and is relevant to a visual cue. So this allows us greater cognitive flexibility to integrate information across domains. All right. So now I'd like to move on to some of the aging effects that we've seen. As I mentioned, there's this dynamic balance between the attention and default network, supporting both external and internally represented information. And it seems as though this dynamic balance really starts to shift with aging. So in this study, younger adults were performing a semantic classification task, and what you can see is engagement of these attentional structures and suppression of the default network, whereas older adults they have this failure to deactivate the default network, that there's not this suppression of the default network during attention to the external space. And it actually, like, the view actually gets a little bit dimmer or grimmer after that. It seems as though with healthy aging, that the integrity of the default network really starts to degrade, that there's a reduced integrity and coherence. And this is looking at both intrinsic functional connectivity, which is on the left and on the right, just during passive fixation, looking at even the higher frequency bands, that there's just reduced coherence of this network compared to younger adults. And this really is not a trivial matter, because the default network and the pattern of deactivation associated with default network is the same as what you see using PIB scanning and looking at beta amyloid deposition in the brain. And beta amyloid is associated with atrophy and metabolic disruption that co-occurs with Alzheimer's disease. So it's not a trivial matter. Like, with healthy aging, if the default network continues to decline, even in the case of healthy older adults, like, this raises a lot of concern. However, some very recent studies have kind of changed this view somewhat. And when you actually look at tasks that drive the default network in older adults, in this case, autobiographical recollection, and another one that came out just a matter of weeks from the previous one, autobiographical recollection and imagining the future, what you see there's really no difference in the expression of activity within the default network with healthy older adults. So this is good news. So when the default network is called upon to recollect or imagine, in healthy aging, these older adults can do these kind of tasks. They're engaging the network robustly and to the same degree as younger adults. But it's very perplexing at the same time. Like, why is it then that this network becomes dysregulated or appears to break down across other types of tasks, or looking at intrinsic connectivity? So we took this planning task as an opportunity to look at the integrity of the default network and the nature of its interactions with both the attention and control network within the same group of subjects, to try to tease this apart. And in order to do this, we had to redefine our stimuli. So we had to find areas where older adults had certain goals that they were interested in accomplishing. That was also consistent with what younger adults were thinking about. So what we did was we interviewed a number of older adults and our younger adult database, and found that there were certain shared goals that both young and old had. And these were prospectively applied to our study group, who we later scanned. And these include scheduling healthcare visits, spending time with family, and exercise. And some of these steps would be, at least for exercise, to make a routine, to walk more, and to avoid injury. These are shared between younger and older adults who could have matched stimuli and compare young and old on autobiographical planning, in addition to the standard Tower of London puzzle. So I'm just going to give a quick glimpse of what the multivariate results look like. This is with partial least squares. And generally what we found was that this method was not sensitive to any aging effects. So what we saw was equivalent engagement of the default and attention networks, and orthogonal to that, engagement of the frontal parietal control network, primarily in the left hemisphere. That was the same for both old and young. But then we dug a little bit deeper, and we specifically associated, we looked at the linear combination of age with the brain activity during the Tower of London task. And what we saw was that the older subjects were, the greater their activity was within the default network during the Tower of London task. So this is consistent with this idea that these older adults were not suppressing the default network during performance on an externally driven task. So they were not pushing the network activity down. However, they were appropriately engaging the default network during this autobiographical planning task. So if we just want to look at what the blood flow is telling us, we see in this area of posterior cingulate, which is one of the core networks of the default network, during the Tower of London task, it's being suppressed in the young. However, it's not shifting from baseline in the old. However, during autobiographical planning, the posterior cingulate is engaged equivalently across groups. Yeah?
Sorry, I just wanted to confirm that older adults tend to have this seeming disintegration of the default network, and yet don't have as much, there's not as much flexibility in it, in attention-driven tasks.
Mm-hmm.
So they're, I mean, I know it's not contradictory, but at first glance, it strikes that they have almost a more robust default network that isn't suppressed.
Right.
But it's, okay.
Right. So also looking at medial prefrontal cortex, you see the similar pattern. They're not suppressing it during an externally directed task, but they're engaging it equivalently as young and old. So it's not actually they're over-engaging it. Of course, this could be an issue of power. These are 18 subjects per group. But from what we have looked at here, there doesn't seem to be a greater engagement than the young, or a lesser. I mean, statistically, they look similar. So taking a similar approach, using these large-scale networks as regions of interest and extracting the bold signal and assessing the magnitude of activity change, what we see in the default network during autobiographical planning in the young is, again, robust engagement and suppression during visuospatial planning. In older adults, again, there's robust engagement of the default network during autobiographical planning, and a non-significant shift from baseline. So again, this failure to deactivate the default network during Tower of London task performance in specifically the older group. In terms of engagement of the attention network, we see an equivalent pattern whereby during autobiographical planning in young and old, the attention network activity goes down, and during autobiographical planning it increases during visuospatial planning. And similarly, as with the previous study, the frontal parietal control network is engaged during both planning tasks in the young and the old. Now, if we look at not just the activity, but the coupling, another interesting story emerges. We see, as before in the young, during performance on the Tower of London task, the attention network is significantly coupled with the frontal parietal control network. So again, on the y-axis, this is the magnitude of correlation between the networks. So here we have the control network, and it's significantly coupled with the attention network during the Tower of London. And the default network is essentially decoupled. In contrast, during autobiographical planning in the young, the default network is coupled with the control network, and the attention network is decoupled. So this is a replication of this task by network interaction. And if we look at this in the older adults, we see during autobiographical planning, there's also significant coupling between the default and control network, and a reduced amount of coupling with the attention network. During visuospatial planning, you see coupling between the attention network and the control network as expected. However, there's much higher coupling between the default network and the control network. And in fact, this is a significant task by network by age interaction. So what seems to be happening, this is not the case that the default network is just more active in older adults. In fact, if that were the case, they would have canceled each other out in this analysis. What seems to be happening is the activity of the default network is tracking with the control network over these tasks. And if we look at whether or not there's a significant difference from baseline, we see we see significant coupling in the expected areas for the young and a decoupling. With older adults, there is not a significant decoupling. So all of these correlations are significantly greater than 0. And what that indicates is, in fact, that there's a reduction in the dynamic range of network interactivity with aging. So just to review these findings, with visuospatial planning, there's a replication that there is a failure to deactivate the default network with older adults. And there does seem to be this aberrant coupling, which I've seen in one other study. And that's where the default network just seems to be more coupled with a control network during task. And it's really not clear what that means yet. However, during autobiographical planning, sorry, did you have a question?
I just, speaking for the beyond midlife crowd here. Is there any possibility these are a failure to decouple or deactivate the default network is portrayed as failure and aberrant? Is there any thinking that it could actually be the basis for something like wisdom? I know I'm fishing here. Keep going.
Sure. There's that. You're right. So those changes in default engagement is not associated with task performance. If we look at high and low performers, so even high performers being equivalent to the young, there's still this lack of suppression of the default network. It's not specifically associated with any neuropsychological measures of cognitive dysfunction. Whether or not it's associated with the more pleasant aspects of aging, the couple that the ones that are known, we're not sure. So the ability to test that is really difficult because we're talking now about individual differences. And then we'd require more than 100 subjects to have the power to look at whether or not that's meaningful. In some studies that have looked at the intrinsic connectivity and seen changes with aging, the breakdown of the network was associated with white matter pathologies and some deficit, but they're not necessarily replicated.
Could still be good.
It could still be good. There is another study that showed that the default coupling during a learning task was associated with greater distraction. Now, that could be that just increased propensity for distraction with aging. And we'll talk about this in the future directions, but that's where it's not necessarily, actually I'll just talk about it in a moment. So when older adults are engaging in the kind of tasks that drive the default network, there is appropriate engagement of this network. And I think that's really a critical thing to see. And that's even in the case of where there's a failure to deactivate. So that suggests that it's not necessarily the default network that's a problem, it's really a failure of regulation. And with aging, there are really two courses for healthy and pathological aging. So with healthy aging, there is kind of a well-noted decline in terms of executive function. However, memory abilities tend to remain intact. It's only when the memory component, when there's memory deficits with aging that are specific to, sorry, let me backtrack a bit. With an age-related decline in executive function, there's often a failure to pay attention necessarily to the environment, and that can lead to a failure to encode information. It's not necessarily a memory problem. Would suggest that the default network can be fine. It's only with pathological aging where the memory problem is more genuine. It's more direct, so that there's a degradation of that circuit. And I'll talk about that a little bit more at the end of the talk if we have time. But again, these findings really do suggest that there is this reduced dynamic range of neural response with older adults, and it's that the young tend to have more of a dynamic repertoire, or more flexibility in terms of how these brain areas are talking with one another. But again, behaviorally, it's not quite clear what this might mean. Okay. And again, in order to understand aging, we're really going to have to see what's driving the default network. Because I think there was a lot of gloom initially when we were seeing this network really just starting to break down and degrade And it's only been in the last year where studies are activating this network. And again, this is an issue of experimental control. People are less likely to do these type of studies where you're having older adults recollect things in the scanner or make these personal plans. And what we see is it's good. It's good news so far. I mean, they can engage this network appropriately, even in the context of when there's a failure to deactivate or this dysregulation. So what I've shown you so far is really a pair of studies where we're looking at these very large-scale brain networks and how they're interacting with each other, but only on the most global of scale. Really what I'm interested in is how are these various regions that we know are supporting different kinds of functions interacting with one another, both within and across the networks. So up till now, it's really been just a gross characterization of the networks, but what's really going to be critical is to understand how these particular regions are talking to one another. And this raises these two questions, like what is a region and how do they talk? So how are they connected? And the question is, like, what is the architecture of the connections between these regions of the brain? And there's a huge endeavor right now in neuroscience, in using fMRI, to characterize what are the functional networks of the brain. And they're using intrinsic connectivity metrics in order to do so. So as time's progressing, we're going from understanding these large-scale networks to really starting to break them down into these discrete subnodes. But one thing that's problematic is that as we're learning more specifically, and specifically these patterns of shared variance, we're losing information about the connections between them, about their functional affiliation. And there's a branch of mathematics that's referred to as graph theory that's really well suited to examine these pairwise relationships between areas of the brain. So this graph theory was really introduced by Euler with the Königsberg bridge problem. And that's what you're seeing right here. This is here we have, in Königsberg, this is the Pregel River, and it's spanned by 7 bridges, and it's connected by 4 land masses. And the question was, could you cross all the bridges only once and arrive at the same point of origin? And what Euler did is he really formalized this question by using nodes as land masses and bridges as edges. And he mathematically determined that, in fact, you cannot cross only one bridge once and arrive at the same origin. So this method of graph theory has recently been applied to the brain to great effect. So in this process, what we do is we can take some measurements of the brain. In some cases, we can look at structural measurements. So some diffusion tensor imaging, where we're looking at the white matter connections between 2 areas, and render this into a correlation matrix that looks really at the structure of the pairwise relationships between them to give us some brain a sense of the network structure of the brain. Another method is to look at time course measurements. So we can select regions of the brain and examine either their spontaneous activity, their activity during task, and correlate this time series across nodes and generate a functional network of the brain. And again, just look at this network structure. But again, this raises 2 really critical points. Like, what is a brain region? Like, what constitutes a node in the brain? In the case of fMRI, what we're talking about is we're collecting signal across 200,000 or so pixels in space. And how do you render something from a pixel to an area that you think might be functionally important or structurally important? And that's not easy to do. And further, like, what constitutes a connection? So in the case of what is a brain area, we can see that there's a number of ways you can characterize space. So in the case of the geopolitical boundaries of the world, if you just took the continents and you wanted to get a measure of sort of homogeneity of function, you can see, like, well, there's reasonable representation of Australia. However, Eurasia is, like, you're going to get a grossly misrepresented average expression of what the geopolitical forces are made of. Likewise, if you just took arbitrarily sized units and spread them out over Europe, or even worse, you did a grid-like function, you're not accurately characterizing the regions that you're sampling. And this is exactly the sort of thing that's happening now in neuroscience with the sampling of the brain. There are methods that are using just fully automated atlases and are just parceling up the brain. And it's known that using arbitrary divisions result in variable network metrics and as well as distortions of the node quality. Just here's an example. This is a very recent study that just looks like Play-Doh almost, how they just separated these various parts of the brain. And we know that these areas are not cohering at all with what we saw with the intrinsic connectivity networks. They were observed using independent components analysis. They don't cohere with task-based activation patterns. So this arbitrary splicing up of the brain and then looking at how they're correlated are leading to poor representations of the network topology of the human brain. And even some simulation studies recently done by Smith have shown that, in fact, it's very detrimental to really accurately portraying how the brain is communicating. So I've taken a very different approach to this and attempt to really functionally localize regions of the brain. So I've now conducted this autobiographical planning study in 3 samples of younger adults, totaling 63 subjects. And what we have here is an average of the activity across the 3 networks during these tasks. And it seems as though we're able to really, first of all, there was very solid, very good replication across the samples. And by averaging across them, we can get a good sense of where a peak functional region is in, say, inferior frontal gyrus or posterior cingulate cortex. And in this way, this is how we started to define, like, what is a region of the brain? Well, it's an area that is functionally relevant and can be specified based upon its task-based activity. In the case of what constituted a connection in this study, what we looked at is we took the bold signal within each of these regions of interest across an interval of time and correlated them. And we used this using intrinsic functional connectivity. So this is where we had our subjects just simply resting in the scanner and perform a number of different pre-processing procedures to kind of clean up the data, because this kind of data is inherently noisy. So in addition to standard pre-processing, essentially regressed out motion, various sorts of nuisance signals in the brain, and did a bandpass filter. So we're really only looking at signal that's between 0.08 hertz and 0.001 hertz to look at these really low scale fluctuations within our predefined regions. And then we correlated this time course across all 43 nodes that we identified in task. And we see that there are possible 903 edges or correlation values ranging from negative to positive. Now, in a standard study looking at intrinsic connectivity like this, usually some arbitrary threshold is determined, either correlation values that represent the top 10% or R values that are greater than 0.2. They're retained and analyzed as part of the network analysis, and all the other correlations are discarded. This didn't really seem like a very satisfying way to proceed. So what we did is we instead relied upon our own sample variance, and we bootstrapped our correlations. So in this case, we had a correlation matrix of the 43 nodes for all 105 subjects. And we bootstrapped those values. And we retained all correlations that were significantly greater than 0. And performing a hierarchical clustering algorithm of these correlation values, what you can see is at rest, the intrinsic connectivity represents very nicely what we saw at task, and that's the control, attention, and default networks of the brain. What I'm particularly interested in this case is really what are these connections that occur between the networks. So in the case of, like, how is the attention network communicating with the control network during, say, Tower of London task, or just how is the brain organized in order to allow that to happen? And how is the default network communicating with the control network? And then between those two hyper-competitive networks, what's the nature of the connectivity between them? So what we see actually is that in any study that uses an arbitrary threshold procedure, they're actually disproportionately removing the between network connectivity. So what these studies are doing are they're representing what's called like a more modular structure. So what we're seeing is where the densest connectivity is within networks, and that's typically what's characterized in the literature. And there's systematically overlooking crosstalk between the networks. So if we look at the network topology of these nodes, you can represent the brain in a very different way. So what you see here in green are the nodes of the control network, in blue are the default network, and in red is the attention network. And this is the lines are the correlation magnitude, and the circles are, the size is a representation called betweenness centrality, which is a measure of connectivity. Further, the shape of this network is actually devised by a Kamada-Kawai energy algorithm. So it's like a Spreng embedding algorithm where propulsive force is set to the correlation magnitudes, and it relaxes back to its lowest state of entropy. So by representing the brain connections in this way, we can actually learn very novel things and compose new hypotheses. For example, the largest nodes with the greatest betweenness centrality have the highest degree of connectivity. And these are these two areas of the control network that support its role as maybe mediating between the default and attention network. And in fact, there's a number of connections from the attention network to these two nodes and from the default to these two nodes, which are in middle frontal gyrus, Brodmann area 6, which is right here. So this is in premotor cortex of the frontal lobe. And this is a region that does two things. First of all, it sits on top of a hierarchical organization of prefrontal cortex. So information tends to enter at this lower system and move forward as things become more complex as we're thinking about them. Further, this area of premotor cortex is also facilitates action. So given the connectivity between default or the attention network, this allows information from the internal system to engage the control network for the facilitation of action. So this is how we can directly behave in response to something that we remember, or respond to something we see in the external environment, can move to a motor command. Likewise, there are other particular nodes involved here where, let's say, the anterior insula is very closely aligned with the attention network. These regions have been associated with the ability to balance, to maintain that dynamic balance between the attention and default network. And what we can actually see, though, is that the connectivity of the anterior insula is very much removed from that of the default network. So prior studies that have shown that this region is critically involved in the suppression of default network, that's actually not the full story, because these regions actually have to traverse a number of different parts of cortex in order to start engaging the default network and potentially suppress it. Furthermore, there are other regions that previously were not well appreciated in terms of their role in interacting between the default and attention network. In particular, something that I'm very interested in is now the left superior frontal gyrus, which is, again, is further up here in the head. This is a very reliable part of the default network. It's always been observed, but it's very rarely indicated as a structure of this network. However, it is very well suited to start communicating with parts of middle frontal gyrus in the control network. So what I'm hypothesizing is that this is a key node to bring internal information to basically conscious awareness, where it can be worked on in working memory, say. And this is the hypothesis that can be empirically tested, and possibly even directly using like TMS to disrupt that circuit. Yeah.
So this kind of builds on a comment you made just now. Yeah. So does that mean that if you found a patient who had lesions specifically to this area 6, that they would have like a particularly disabling set of symptoms?
It's possible, definitely.
Is that something that's been found? Because usually when people talk about the patients who have really bad planning problems, or have some more kind of frontal.
Mm-hmm.
So what happens to someone? Presumably there must occasionally be patients who have lesions like this.
Right. A lot of the planning deficits, like with Tower of London, where we're seeing our deficits as a result from these the lateral frontal areas. So here and here as well. And indeed, it could be a consequence of not being able to bring information online, or a function of that specific node. There's some really interesting work that Mark Esposito is doing. He's turning now to look more at graph theory metrics to characterize brain injury. And interesting, what they're finding is it's not just a specific region of the brain when it's damaged that there's a loss of function. There's actually a perturbation across the entire network that causes dysregulation. So that it's not, that local deficit actually has very global problems. And they tend to track along these large-scale networks too. So this is one way that this method is capable of really characterizing the brain in a very different way and can answer like very unique questions. So how are we? Do you want me to talk about future directions, or do people have questions? All right. So I'll just tell you very kind of briefly what it is that I've been thinking about and working on. And I mentioned this earlier, how there seems to be these two tracks that change with age, and that's with healthy aging, there tends to be more of a specific decline in executive function. And it's really only with pathological aging and Alzheimer's disease where you have a degradation of the memory network or the default network. Now, a lot of the work I've been showing you is really how these two systems are communicating with each other. And one of my hypotheses is that looking at these, we've learned a lot by looking at these domains in isolation. But I think that we're really going to benefit from looking at how they interact with each other. And it could be that we might see perturbations or disruptions in the connections between these networks. They might be more sensitive to disruption at an earlier stage of disease than just looking at within in the individual domain. And there are a lot of tools now that are opening up that allow us to do these sort of things, not just at task, but also looking at rest. So looking at these intrinsic oscillations in the brain, we are starting to really get a good sense of how to characterize the integrity of these circuits without necessarily having to drive them by task. So in a recent study that came out of WashU, they've done a better job than I have of parcellating the entire brain into functional units. And they're able to span many different networks. And what they're moving towards is this ability to use a functional atlas of the brain. In this case, this is partially based on large-scale meta-analyses, looking across hundreds of different types of tasks, and combined with resting state connectivity and looking at shifts in connectivity patterns, just to get a map of parcels of cortex that are functionally distinct. And in this way, we can take these maps and apply them in novel ways and basically redefine how we think of brain areas and look at the nature of the interconnections both in health and in disease, and see if we can really start to predict these states. And we can do this not just functionally, but also structurally. So there's a kind of an old adage that neurons that fire together wire together. So when you tend to have this co-activation of cells, they become connected. They form a network. Well, in fact, what happens is like these cells that fire together and wire together, they also die together. So in the context of pathological aging and dementia, with Alzheimer's disease, frontal temporal dementia, cortical basal degeneration, where you see are not just one area start to decline, but all these areas that are connected to that area are declining at the same time. So you're having a reduction in tissue integrity. So we can actually start to see not just functionally, but just structurally, changes in the network integrity in health and disease just by looking at the structure of the brain across a number of different older adults. So this is some work that we'll be spanning over the next few months, kind of in anticipation of the scanner. I've actually started analyzing other pre-existing data sets that are currently out there. We can look at hundreds and hundreds of scans and to assess network integrity on this basis. So these are projects that are ongoing, and in addition to a whole wealth of other things that I'm going to be talking about on Cornell Neuro Day. So if you want to come to that, I'll share with you the work I'm doing in social neuroscience and kind of the intersection between autobiography and social cognition and how they can inform each other and the relationship to the brain. So thank you very much for listening.